Hi, I'm Hugo, an engineer and a future PhD student in Vera Pocaldi lab at City in Toulouse. And I'm going to make you a brief presentation about a project called Dissecting Cellular Communication through Gene Regulatory Network Inference. So we currently explore the tumor microenvironment in the lab, and we know that macrophages can be educated by the cancer cells themselves to promote tumor growth, becoming tumor-associated macrophages that we also call TAMs. These are located within the tumor microenvironment along with other kinds of human cells. Monocytes can be recruited and differentiated into macrophages that contribute to the formation of NLC or TAM, M2-like macrophages that are anti-inflammatory and that will protect cancer cells. So CLL is a slowly progressing cancer that starts from white blood cells in the bone marrow and extend into the blood. This disease is characterized by an increased production of mature but dysfunctional B lymphocytes. CLL emits cytokines that turn monocytes into NLCs and are attracted into by NLCs. So in terms of data, we have three different conditions. The first one is heterologous culture containing monocytes coming from the blood of a healthy donor and B cells coming from a patient having CLL. We also have two other cultures, one autologous and one B cells. The autologous one containing all the PBMC, and the B cell have been isolated to form the monoculture from the PBMC. The main objective of the project is to study the different interaction that occurs between the human cells and all the cancer cells. For that, we build networks of interaction between all these different genes and different cell types in the tumor, healthy and cancerous cells. And at the end, the main goal is to build a multi-scale dynamical model. So the first step is to infer the regulatory network from the CLL data. For that, after some pre-processing step, we use DNGenny3, an algorithm based on random forest to infer the gene regulatory networks. And following that, we performed some analysis like topology, gene set enrichment, and some kind of centrality measures. And at the end, we obtain three different networks like this one for the three different cultures. And in this network, we have like the genes that are involved in the three different cultures. And we can observe the most central nodes based on different centrality measures, depending on the color, the size, or the shape of the nodes. To complement this gene regulatory network inference, we also use TCSEQ. TCSEQ is a package for analysis time course sequencing data, and it can be applied to transcriptomic data and epigenomic data. It focuses on differential analysis between different time points and temporal pattern analysis. In our case, different biological processes have been identified, like dynamic changes in immune cell population, cell proliferation and differentiation, and also, like this one, a metabolic shift. Here we have a decrease in reliance on glucose as an energy source as the culture progresses, and increase in lipid metabolism, or we can call that also an adaptation to nutrient availability. We complete this analysis with some CNET plots to link the different biological processes to the TF or gene associated. The last step is a validation step, one of the most difficult, I think, in our projects. And for that, we use both co-expression here with the string network and motif using the RSys target library. In conclusion, inferring gene regulatory networks remains a challenging task, more particularly with the selection and validation of inference method. This is the main obstacle. However, data-driven GRN inference, especially in the context of RNA sequencing time series data, show promise for identifying novel therapeutic targets. Developing dynamical models for disease like CLL can provide valuable insight into cellular regulation. And despite ongoing challenges, data-driven approaches offer a powerful tool for advancing our understanding of gene regulation and possibly precision medicine.